77 and a 76. That's following a 3-question adjustment. I did find a couple questions I wanted to just give everyone a point for because the performance was on the lower end. They were doable, but the performance was a little bit rough. And then I just threw in one more for good measure. So the 3-question adjustment in the
Yes! Oh my fucking God!
So exam viewing recommences now. We close office hours after class. Stop on by and do your exam. We'll continue through next week. Right? So in 2 weeks, get 2 full weeks of your exam. I strongly encourage you to do that. If not, to learn about metabolism, maybe address questions about how to better approach exam items and approach your studying, which is before the exam takes place. These are questions, remember, you can bring to the table. You can ask your TAs and they'll be happy to help you. Okay, I think enough of that. Let's go ahead and continue. So we start our final unit, or rather continue, because I trust you looked at the posted video lectures for last Wednesday, right? So the topic that we start off with is cell division. From here we go on to some classical genetics, and then we round out the course with molecular biology. So this last unit is very much about the genetic material, about the DNA, right? Why we have it, what we use it for, and what we need to do with it to keep life going. Remember we said life is transient, as you know well, but we continue, life continues, it goes on because of the capability of reproduction. Right? So we talk about all these things here in this last unit. And it starts with chapter 12 and 13, which deal with cell division. Right? So nothing new here, and I hear you guys are eager to go on, so I'm not going to just kind of say the usual. But notice that you do have some assignments posted. If you haven't had a chance to check Canvas, the mastering assignment is over chapter 12, right? Not 13, over chapter 12. And we're going to finish chapter 12 today. So once we've done that, after today's lecture, you can go ahead and complete the mastering assignment over mitosis, okay, without further mystery. Please finish that by Wednesday, okay? There is a quiz as well over both mitosis and meiosis, which we're starting today, meiosis, and we'll conclude that on Wednesday. So after this week, you'll be in position to complete that quiz to Saturday. Okay, so these are the assignments that have been posted. One important heads up about the quiz, it does have a worksheet, like all the quizzes do. You should complete the worksheet before taking the quiz. That said, if you do not do that, you will fail the quiz, right? So, you hot dogs who like to go ahead and just, I'm just going to take it anyway, no, because the questions won't make sense. The questions are in reference to a figure that's only available in the quiz. Okay, so look at the quiz worksheet and read it carefully as you complete the worksheet. Carefully, read it carefully, because I think, I know actually that we have a tendency, maybe as Homo sapiens, to imagine that every question about biology is about Homo sapiens. Well, that's hardly the case, because life is hardly about Homo sapiens, right? We're not the only type of life, we're not the pinnacle of life, right? We kind of think of that way, like everything is about us, Homo sapiens. Yeah, that's very narrow view in my opinion. Okay, so look at this quiz worksheet very carefully and recognize what it's dealing with as though it's not Homo sapiens. Okay, and then if you complete that, I think you'll do well. Intervention number 6 is the last intervention. I'll post that after class today. Please complete that by Saturday. Okay, that's it. Any questions about anything before we get started? All right, I think again you are eager to get going. So let's go ahead and pick up where we left off. Again, you are responsible for the content of the video lectures. If you haven't looked at them yet, please quickly do that because you're now out of sequence with the lecture content. One of the things that was covered, it was a lecture over the genetic material. Again, over DNA and the various terminologies that we utilize to talk about DNA and what it is and where it is and what what it looks like, and there's a lot of jargon here that deals with DNA. And yeah, that was pretty much, I think, the lecture in a nutshell, okay? So Leslie, one of the things we did say was that eukaryotic chromosomes, right, will change in terms of their structure, right? They're going like this. Their structure, that means how dispersed they are in the nucleus of the eukaryotic cell, right? Eukaryotes all have a nuclei in the cell, and nucleus, right? Called the nucleus. It could be spread out in the nucleus, but it could also be condensed, right? This is the word you hear, condensed. So more or less condensed. And this is a fundamental aspect of eukaryotic chromosomes, which I suspect you don't know a lot about, if anything. Okay, so that, please understand that, and we'll see how it's important as we talk about mitosis today, right? Now, what is a eukaryotic chromosome? Well, it is one DNA molecule, right? So by definition, a chromosome is a single DNA, and a eukaryotic chromosome is a single DNA, or one DNA molecule. What is its shape?
X.
Linear. Yeah, okay, I did hear X shape. I don't like that answer. With respect, and I think I know if you said that, or if you think that even, I know what you're thinking, but I don't like that. And there's multiple reasons I think can lead you down a dark path if you say, oh, chromosome is an X, right? So, I was going for linear. Remember that DNA can be a single linear chain, right? Or they can be what? Circular, right. So bacterial chromosomes are usually circular. All eukaryotic chromosomes are linear, yeah. So this is a thing now, guys. Yeah, everyone knows eukaryotes have nuclei, right, and their cells, and bacteria don't, prokaryotes don't, right? That's nothing, right? Oh, nucleus, eukaryote. No, what does it really mean to be a eukaryote? What are all the differences that you don't hear when you just hear eukaryote, right? That's what we're getting into here. That's what this is about. So the DNA is fundamentally different from bacterial DNA. It's a linear DNA. There's consequences to that. There's consequences. It's actually difficult to manage. More difficult to manage linear DNA than it is circular. It may not be intuitive, but it's true. Okay? Moreover, a eukaryotic chromosome, eukaryotic chromosome is a DNA molecule as well as protein. All right, now that's important. And that's really, I think, again, more speaking to eukaryotic chromosomes than bacterial chromosomes. All right, so why is the protein important? Well, that is, in a nutshell, why the DNA can become, or the chromosome can become more or less condensed. It's because the DNA is wrapped around protein. Right, it's wrapped around protein. A chromosome contains protein because the DNA is wrapped around protein. Okay? What are those proteins called? Histones. A couple of you know that, a couple of you, and the rest of you don't. That's fine. I didn't cover that. Or I might have just said it in the audio lecture. I'm not going to ask you that question. Not in this course, but that is true. The protein is called histones. All right. Moving on. Before cell division, which is, of course, the process that gives us more cells, right? Again, fundamentally linked to reproduction, is the ability to make more cells. Before cell division, a chromosome is what?
Replicated.
Replicated. Yeah, that's the answer. And I think it's the best answer to that question. Replication is a term that specifically describes what happens to a chromosome and what happens to DNA in particular. Replication. DNA is replicated. Okay, very good. Now that, I don't know, maybe it's intuitive to you. Great if it does. This is profound. is profound. There's no cell division that I know of that is not preceded by DNA replication. These processes are linked. And if you're doing binary fission, it's preceded by DNA replication. If you're doing mitosis, it's preceded by DNA replication. If you're doing meiosis, it's preceded by what?
Replication.
No. It's Monday, I get it. Come on. If you're doing meiosis, that would be a type of subdivision. It's therefore preceded by what?
Replication.
DNA. No. I'm about to leave here. This is ridiculous. Folks, every cell division is preceded by what? DNA?
Replication.
Meiosis is a type of cell division. Therefore, it is preceded by what? DNA?
Replication.
Replication. Okay. I'm not convinced, but I think we gotta go.
That was a pressure out.
That is what messes you up. Forgetting that. If you're, I'm not sure, meiosis, it gets confusing for me. No, not if you remember the DNA replication. Okay, profound thing that happens before cell division. Okay, I don't know what else to say. So a replicated chromosome, then we have to think. A chromosome can be a chromosome as we define it here, or it can be a replicated chromosome, right? Which is the product of this process, DNA replication. A replicated chromosome would consist of two what? Okay, I hear some different answers there, and that's actually appropriate because there could be more than one answer. So in terms of DNA, if a eukaryotic chromosome has a single DNA, but now we replicated the DNA, how many DNA are in a replicated chromosome? 2. Yes. A replicated chromosome has 2 DNA. Exactly correct. The other, I think I heard the other answer is sister chromatin. Because when you look at a chromosome under the microscope, you can't see DNA. Right? We'd be lucky to see the chromosome, but if you look at the right time, and of course have a microscope, you could see a something, right? And you could call it a chromosome, and then you could say, yeah, I think it has 2 of something, and we call it a sister chromatin. Okay, there's a figure in your text that shows all of this, actually. Like everything on this slide is on this one figure, which again, I'm going to say it, this is a great one. This is one you spend 20 minutes looking at this. You think you're done, and you're still not. Okay, because you haven't really, Okay, so let's take a look. Let's look at it together. So clearly a process, right, from top to bottom here. And this is a simplification, right? Because eukaryotes typically have how many chromosomes in their cells? Okay, see, now you're Homo sapiens again. Don't tell me 46 chromosomes. Who said that? Homo sapiens centric? Okay. Okay, so, yeah, that's true for us, but not most species, right? That's our thing. Some have more. Some species have more than 46 chromosomes, and some have less as well. But it does make a point that many chromosomes. Okay, so eukaryotes typically have many chromosomes, not just one. Okay, you got to roll with the simplifications here, but that is what you're seeing here, just to keep it simple. Okay, what is a eukaryotic chromosome, or what does it contain? A single linear DNA molecule. Okay, so that's shown here. There is that DNA. So what is that? Well, it's going to have that in it. But then it also has protein, which is what allows it to achieve this state. They're showing the condensation, I think is what they're going for. It's a little bit fictitious there as well, but I think I won't say anymore. Okay, so the point is, before cell division happens, what happens before? DNA replication. That's right. There it is. Okay, so cell division is yet to happen. It hasn't happened here, but DNA replication has, right? And what does that do? It makes one DNA into two. It copies the DNA replication. It copies the DNA. And then you have this, which is what everybody looks at and says chromosome, right? Oh, yes, it's a big chromosome. Yes. More accurately, it's a replicated chromosome. You say chromosome, but more accurately, it's a replicated chromosome, right? It's a product of DNA replication. I know it doesn't say it there, but you know, okay? So yeah, that's what gives us that thing. And that means that's not one DNA, it's two DNAs, right? Replication, two DNAs, and chromosome. Cell division will separate those replicated DNAs, right? So each cell will get one of those DNAs and have it. Each cell will have the DNA the original cell had. Okay? So achieving that means separating the sister chromatids. Right? That's what mitosis does, separates sister chromatids. And that's it. This is really mitosis and DNA replication all in one. Okay? Question? Before we get into this? No? Okay. All right. Let's go ahead and talk about mitosis. I hope the little bit of review there helps you get back into the results. All right. So let's begin with the question: what does the cell use to distribute sister chromatids in the course of cell division? I mean, like physically, what is it? How do you distribute the sister chromatids? What does this thing with my fingers? What is this called? Isn't this a spindle?
Yeah.
Maybe not very well demonstrated, but that's a spindle, right? That grabs the. What is the spindle? Where does that come from? What is it made of?
Protein.
If a protein, yeah, is a good answer, I think, because protein does almost everything in the cell. Like almost everything is protein. It is protein, actually, but specifically it's the cytoskeleton. So now remember what we established at the end of chapter 6, actually. We said the cell has a skeleton just like we have a skeleton, and that skeleton gives shape and support to the cell, but it also can change shape. It's also dynamic, and the dynamic quality allows it to be repurposed into things like the spindle that distributes sister chromatids during cell division. Okay? So cytoskeleton is front and center in this process of cell division. Maybe something else you were not aware of. Okay? Cool. So we're going to now look at mitosis, and we will do so with this stunning collection of micrographs, fluorescent micrographs, which show a cell dividing. So what are we looking at? You are looking at a cell, but only some portions have been stained. So I'll tell you one. Or you tell me, what is that? Nucleus, I heard nucleus, right. So I think it's a reasonable answer. Technically, it's the DNA. The DNA has been stained. Where's the DNA? In the nucleus, right. So there you can see the nucleus because the DNA has been stained. So now we have to kind of focus on the DNA, see what's going to happen in the course of cell division. What's all this? Outside the nucleus. I'm going to ask you. That's a cytoskeleton. Yeah, someone say that? Yeah, that's what that is. So there's two things that have been stained. The DNA and the cytoskeleton. So you can follow the fate of both of these in the course of cell division. Right? You do notice that some of the cytoskeletal fibers meet up here. It's not that they're meeting up, actually they're coming out of, I think that these fibers are coming out of this structure. What is that? This is an organelle we didn't talk about called the centrosome. Centrosome, or central body. Right, it is sort of in the middle of the cell, isn't it? You think of that, yeah? Centrosome. The fibers, cytoskeletal fibers come out of that, particularly microtubules. Remember, microtubules grow out of the centrosome. What species is this? It's not really, you didn't think human, did you? Be careful. Again, we're not the only kids on the block. This is actually a newt cell, not a human cell. And I think the reason they chose it is because it has less than half the chromosomes of a human cell. Yeah, and we do have 46 chromosomes, someone said earlier, and it gets kind of confusing trying to follow the steps of cell division with so many chromosomes. It'll still be confusing with 22, but at least it will be a little simpler, okay? These micrographs are accompanied by diagrams, so you can see clearly what you're seeing in the micrographs are incredible. These are just beautiful work, but still hard to see maybe individual details, and so you see that here. Okay, so I think this is maybe, yeah, so what's going to happen, we have to see it. But I didn't mean to do that right there. So let's look at the cell before cell division, because this is what we're looking at here. This is actually G2 of the cell cycle. I presume you remember what that is. It's near the end of the cycle before we get into what? M phase, nice, right. When mitosis happens, right? It's before that. So notice that at this stage, G2, or phase, the centrosome has been duplicated. That's important. The centrosome has been duplicated. The centrosome from which what grows out of? Microtubules, okay, has been duplicated. So that's important. Another thing to recognize is that the DNA is very spread out. We talked about how it can be condensed. This is decondensed here. This DNA is decondensed. And in a decondensed state, remember that chromosomes have a name. We refer to them by chromatin. Yeah, that's when the DNA is decondensed. Chromatin is just the DNA protein complex. The complex of DNA and protein bound together. Chromatin. So chromatin we have in the nucleus and 2 centrosomes, and yeah, that's it. Okay. So now we'll proceed to talk about what happens to that chromatin and those centrosomes in the course of mitosis. Right? Remember what that is. Mitosis is the division of the nucleus, right? So when a cell divides, the nucleus or the contents of the nucleus is also divided up between the daughter cells. And so what happens there is referred to as mitosis. I mean, as you know well, mitosis is further divided into phases, right? Various phases of mitosis. And you had to memorize these at some point in the past. Once again, you do need to know them, but also recognize, if you didn't before, that these names are very intuitive. Okay, we'll talk about mitosis as it is occurring through 5 phases, and these names are intuitive. Pro, phase. What does pro mean? Before. Yeah, before. Prokaryote came before. Eukaryote. Prophase is the phase before all the other phases. And prometaphase, obviously, happens before metaphase. What does meta mean? Yeah, middle. One meaning is middle. Middle. Meta means middle. That's when the chromosomes do what?
In middle.
Line up in the middle of the cell, of course, but it also happens to be the middle phase, and that's kind of helpful as well. Ana means apart, so anaphase is when the chromosomes are pulled apart. What does kilo mean? End. Well, good. A kilo means end, and that's the last thing. Okay, good. So, of course, it helps to know those prefixes, I guess. What do you think? Okay. So once again, we're going to start at G2, but then proceed into M phase, right? So watch for what happens. In G2, found in the nucleus is what again? Chromatin is the best word. Are there chromosomes in the nucleus here? Is that correct to say? It is correct, actually. Be careful. It is correct. Okay? I gotta think of that as a question, because that's a good question I could ask, right? I'm serious. It's not on the exam, but it's not about name, right? But is it correct to say it was chromosomes in G2? What was the answer? Yeah, they're there. You just can't see them. Right? They're spread out in that decondensed state of the DNA, and you can't see them, but they're in there. Of course. Of course. Chromosomes are always in our cells. Of course. Right. But in this state, they're best referred to as chromatin. You're looking at that, you can't say chromosome, right? Because that's not, what is that? I mean, that's part of a chromosome, I guess, but it's chromatin, right? That's how you, does that make sense? Okay, working with the jargon, folks, understanding the wording is everything in cell division. So you gotta make your peace with this, okay? All right, so beginning with prophase, big surprise, in prophase, the chromosomes condense. Yeah, they're decondensed, but in prophase, beginning of mitosis, they condense. So let's say it now. One reason to condense DNA is to manipulate it, right? To grab hold of it and pull it apart, which is what the spindle is going to do, right? So how do you do this unless the DNA is grabbable, right? And resilient to manipulation. Chromosomes condense. This is very intuitive. So apart from the naming being intuitive, what happens when is super intuitive. I think you really have to be working to mess this up. Given adequate time and preparation, there's no way every single one of you cannot master this topic. This is very doable. Another thing that happens, though, is that the centrosomes begin moving to opposite ends of the pole. Opposite ends of the cell, right? Opposite ends of the cell. And again, centrosomes are what shoot out what? Microtubules, part of the cell skeleton, right? And that's important because that's what we're going to do, right? We're going to build the spindle from microtubules. How are we going to do that unless the centrosomes are spread out? Okay, so this is again intuitive. Right at the beginning, you want to begin spreading out the centrosomes and beginning to form the spindle. So right away, I think the microtubules begin to shoot out, begin to, kind of form that structure, which will ultimately call the spindle. All right, now the idea is that the microtubules grab hold of the chromosomes, of course, to pull them apart. What is a problem to the microtubules grabbing hold of the chromosome at this moment? Okay, so you gotta figure out which one to grab, where to grab, even. It's a little, it's correct, but it's too complex. Think simple now. Why can't the microtubules grab the chromosomes now? down. Another word for that is disintegrate, right? Disintegrate, come apart. Good word to use. Because things that break down or disintegrate could conceivably reintegrate, right? Yeah, okay, good word. So that allows then the microtubules to grab the chromosomes, right? So as soon as that nuclear envelope is gone, you can make contact now. And where do you grab hold of a replicated chromosome? At the what? Centromere. Wow, very good. Okay, you do know this. You grab hold at the centromere. The centromere. Okay, so remember, the centromere is that narrow waist of the replicated chromosome. Okay, I know you know what I'm. Okay, so if this is the chromosome, right? This is the centromere. Okay, that's where it grabs hold. Very good. And then we go to metaphase, which is where the chromosomes are what? In the middle of the cell. Right, so after we grab hold of the chromosomes, we can move them around and they begin to move back and forth and get them line up in the middle of the cell. Okay, so the microtubules pull them into the middle of the cell. And I think this has been described as a tug of war. It's very apropos. It's like a tug of war, the two centrosomes pulling on the chromosomes via the microtubules, right? So, tug of war, if you will. Well, let's take a look. What was the first thing that happens in prophase again? The chromosomes condense. You can see that. You can see that in this micrograph. This is ridiculous. Look at that, and look at that. Already, we can see something happening. And what's happening is the chromosomes are condensing. The chromosomes that were there all the time, all along, are now becoming tangible, right? They're becoming visible to a naked eye, right? Or naked eye with a microscope. Okay? All right. But, and the centrosomes begin moving, and they begin shooting out the microtubules, right? But in order for the microtubules to grab the chromosomes, you have to get rid of the what? Nuclear envelope, prometaphase. Boom! What happened from that to that? Clearly, the nuclear envelope was lost, and suddenly the chromosomes are spilling out, and they're out in the cytosol, right? And the microtubules are grabbing hold, right? That's already happening. So as that envelope disintegrates, the microtubules grab hold. Now notice that while some microtubules grab hold of chromosomes, other microtubules grab hold of each other. Everybody see this? Okay, some microtubules grab hold of the chromosomes. There we go. Centromere, centromere, centromere. But some microtubules grab hold of each other. Yeah, good diagram. Very clear. All right. There's some terminology here, which I think I'll just let it hang. I do want you to know it, but I'll specify why this is named as such. Let's just look at this a little more closely. Okay, so every replicated chromosome has a narrow portion, which again is called the centromere, right? Careful you don't confuse it with centrosome, obviously. Obviously. I wonder if he's going to ask us that. Yeah, I'm going to put that. It's like given, right? Centromere, centromere, telomere, right? Anyone know that term? Telomere, the ends of the chromosome. Telomere, centromere. Okay, so the microtubules grab hold of the chromosome at the centromere. Understand that at the centromere, for each chromatid, There's, where's my, oh gosh, I thought I had a label like that. Okay, there's a complex of protein. Let me say it again. For each chromatid at the centromere, there's a complex of protein. That's called the kinetochore, or kinetochore complex. Kinetochore complex. Okay? And that is actually what the microtubule got hold of. Okay, so how does a microtubule grab a centromere? It's because of these kinetochore complexes. They're attached to these kinetochore complexes. They're just a complex of proteins that's really sticky for the microtubules. Okay? And you can look at the micrograph again to see what's happening. So these microtubules are going to pull on the chromosome, having attached to the chromosome, going to pull on the chromosome and line it up in the middle of the cell. And when it's all lined up, when all the chromosomes are lined up, we call that metaphase. Yeah. Right, this is a continuous process. I should have said this earlier, but the reason I put that animation in there, that kind of goofy animation, is because it's like, whoop, what happened there? Whoop, whoop, and again, whoop, it's a continuous process. And we have spent hours and hours under looking at the microscope to find these phases, right? To break it up into parts, but it's just a continuous process. So in order to understand it, we say, okay, prophase, prometaphase, metaphase, anaphase, telophase. All right, metaphase. That would be metaphase, they're lined up in the middle. Question? Okay, no? Anaphase, what happens? Chromosomes pulled apart, yes, of course, because now we're ready. Now we've pulled them apart, now we're ready, we've lined them up, we're ready to pull them apart. So sister chromatids are pulled apart. Sister chromatids, I know you're not writing down what's on the slide, you know you will write down what I say. Sister chromatids are pulled apart, which of course is also the chromosome being pulled apart. Now also understand that in anaphase, the microtubules attached to each other begin to do something. Okay, so we need a name for these microtubules because clearly there's, again, some are attached to the chromosome, some are not. The ones that are attached to the chromosome are called kinetochore microtubules because that complex, again, is called the kinetochore. So the ones that are attached to each other, we just call non-kinetochore microtubules. Microtubules attached to each other, non-kinetochore microtubules. The non-kinetochore microtubules begin pushing on each other. They begin pushing on, the non-kinetochore microtubules begin pushing on each other. And that pushes the centrosomes apart. That pushes the two centrosomes. So we don't really see centrosomes anymore at this point. The centrosomes have become the ends of the spindle, right? And so we call them spindle poles. So centrosome is a term that's more appropriate to those organelles in the interphase cell, but during cell division, they become spindle poles, okay? And so the poles move apart because of the non-kinetochore microtubules. The poles move apart. In telophase, so that's it, right? You pull the chromosomes apart, you move the centrosomes apart, and that's going to elongate the cell. That's going to elongate the cell. That's what it says here, right? Elongate the cell. You're kind of done now. You just need to wrap up. One more phase to wrap up. All right, so what's going to happen to all of the little pieces of nuclear envelope? Remember, it disintegrated. What's going to happen to those pieces? How about reintegrate? We've separated the chromosomes. How about reintegrate into new nuclear envelopes? At which point, nuclear envelopes will reform from the pieces of the old one. Okay, again, it reintegrates. And then the chromosomes decondense. Okay, so let's look at all that. So here we were in metaphase, and we said in anaphase, pull apart the sister chromatids. Again, the kinetochore microtubules do that. While at the same time, the non-kinetochore microtubules begin to push apart. They push apart, pushing the spindle poles apart and elongating the cell. So can you see how the cell is longer now? Now, they went for that versus that. It's becoming longer. That's also important, right? So in telophase, again, nuclear envelopes reform. Kind of hard to see that happening there, but it is in progress, I believe. Right? There you go. There we go. There we go. And the chromosomes decondense. And then we got to make a point that the cell itself splits in two, right? So that has a name. We call this cytokinesis. The cell splits in two. I'm just going to call it cytokinesis and we're done with it. Okay? Cytokinesis. Cytokinesis is the cell splitting in two. And I think your text gets into the mechanical aspect of that. Question? Yeah. So again, it's very interesting. So he's asking about, what is the distinction that goes on between the kinetochore microtubules and the non-kinetochore microtubules. Okay, it's been a little while. Let me see if I can get all this. So what happens with the kinetochore microtubules is they pull apart the chromosome. So it's pulling, right? Pulling apart the chromosome. And that really requires them to become shorter. So these microtubules begin to shrink, and that creates that pulling action. At the same time, you have the non-kinetochore microtubules growing. So they're becoming longer, which moves the poles apart. And I believe there's action happening here as well. So these are growing longer, but they're also kind of moving apart. Like that, you know what I'm saying? So, just like that. And so, a lot of, bottom line, anaphase, there's a lot of sort of counteracting forces happening, right? Counteracting forces that pull apart the chromosomes and push apart the spindle poles, which elongates the cell. Okay, pretty clearly, that could be a whole lecture in itself. Very good question. Very, very good. Okay? I just will conclude by saying that cytokinesis is also done by the cytoskeleton, right? But not the microtubules now, the microfilaments. Remember, microfilaments form contracting structures that contract, like your skeletal muscle, yeah? That's all microfilaments that's causing the muscle contraction. Likewise, a contracting ring of microfilaments is what pinches the cell in two. So let's say that their cytokinesis happens after telophase and involves a ring of microfilaments and divides the cell in two. Okay? All right. Anything else? Yeah.
If we were on the test, would you have those on the test, or would you have the other ones that clearly show?
So I can tell you, so the question is about what will you see on the exam with regard to images. So no and no. So definitely not these because I don't think they will copy well, but also not maybe one figure of a dividing cell. I'm not really going to, here's the cell, label the parts, but I'm not saying I'm not going to do it. So I would expect one or two maximum figures, okay? But more, I think, be kind kind of cognizant of what's happening when, so for questions, we're just, you know, in written words, you would be able to say. Okay? Yeah, good. What else? Okay, if that's it for the moment, then let's go ahead and begin to talk about that other type of cell division, of course, being meiosis. I'd like to, as we did with mitosis, just set the stage. Okay, we don't do meiosis today, but let's just set the stage for it, because like mitosis, you really have to understand quite a bit about the genetic material before diving in. Okay, so let's talk about sexual reproduction, anyway, a little bit. Recall that bacteria divide by a type of cell division. It's not mitosis, it's not meiosis, it's called binary fission, right? So this is kind of the beginning of cell division, the early stages of life, right, or history of life on Earth. Okay, then come along eukaryotes, and eukaryotes today can reproduce. I mean, for bacteria, fission is reproduction, right? One cell splits in two, you've reproduced. For eukaryotes, it could be that simple, but it could also be more complex. Okay, reproduction could be just a cell division, but it could also be more complex. So, long story short, I guess there's two types of reproduction, remember? Right? So what are the types? Sexual and asexual. Okay, so you know that, right? So what are they? Well, we just listed them. How are they different? So sexual reproduction obviously involves two parents. Then what is asexual? One parent. That can happen. One parent produces offspring. It's called asexual reproduction. And then how is cell division involved? Okay, well, let's start with asexual. So asexual, I would say, is more of a mitosis, right? So you could be single cell, and then you divide, and now you reproduce. So just like the bacteria, the cell divides and you reproduce. And that's going to be more of a mitosis, kind of a mitotic division. Although there are multicellular eukaryotes that also use mitosis to reproduce. Okay, so I think the hydra is your example from your text. The hydra, the sea hydra, is multicellular, but one individual will produce, it's producing an offspring here for my shoulder, right? It's called budding. Budding, it buds off an offspring, again, using a mitotic cell division, okay? So reproduction could involve mitosis for some of these simpler forms of eukaryotic life, okay? Of course, then you have sexual reproduction, right? So you have eukaryotes, ostensibly more complex eukaryotes, which are using two parents to reproduce. Two parents are necessary to produce offspring. And in that case, meiosis is involved. Meiosis becomes involved in sexual reproduction. Okay? Is that clear? Okay. So that said, and kind of contextualized, meiosis is a very special kind of cell division. So again, only sexually reproducing eukaryotes do it. Moreover, it only happens in one part of the body. It doesn't happen here or here or here, right? It's only in the gonads, right? Gonads. In female, what do we call the gonads?
Ovaries.
Ovaries, yeah, it only happens in the ovaries in female. In male, we call the gonads testes. It happens in testes in males, right? It happens in both females and males, but it only happens in that location, ovaries and testes. And it produces a different type of cell that's different from all of the other cells of the body. Okay? All of your body cells are literally called body cells. They're called somatic. Again, som meaning body, right? Somatic body cell. So a gamete is a reproductive cell, right? It's for the purpose of sexual reproduction. And so it's a different type of cell. So you have all the cells in your body which are somatic, and then you have the gametes, which are the product of meiosis. Okay? So where's the only place in the body you would find gametes? In the gonads, yeah, because that's where meiosis happens. That's the only positive that will give you that. Okay, very good. So what is different about gametes, the obvious, right, is they have half the number of chromosomes. Gametes have half the number of chromosomes of all the other cells. Because meiosis does that. Meiosis halves the number of chromosomes. And of course, the purpose is not to stay that way, right? The purpose is then to reunite, or not reunite, but to bring together gametes, right? To bring together a male and a female gamete to create a new individual, right? At which point you restore the original number. Okay? So the unification of male and female gametes is called fertilization. Okay, sperm fertilizes egg, right? Fertilization, and that will restore the original numbers. Now this is very complicated. I mean, again, compare that to this. Right? Making an offspring. Why not just do that? Why not just have one parent? It can happen. It does. What advantage could having two parents give you?
Genetic variation.
Genetic variation. Yeah. Yeah. So there's an adage in evolutionary biology, right? So things that persist over time probably persist for a reason, right? There's an advantage. And if it's a little time, that's one thing. Let's see. If it's a long time, then it really argues again for an advantage at some point. So the advantage here is a very, very complex type of cell division and mechanism of unification, right? A lot of these can go wrong. There must be an advantage. The advantage is variation. Variation. Right? I think people say genetic variation. That's true. But let's just say variation. You get more offspring. You get a lot of offspring. And different kinds of offspring. Right? Different types. And that's good. Because if one offspring doesn't survive, then maybe another one does. Right? Variation is everything in biology for reproductive success. Okay? Any questions? All right, maybe at the end of the context there, let's talk about chromosomes once again. And this time we will look at human chromosomes, okay? After lecturing you on the Homo sapien-centric attitude, let's take a look at human chromosomes. And there's a old classic technique by which you break open a cell, take out the chromosomes, and then lay them all out and study them. Okay? So you have to get a dividing cell to do this. You have to get like a mitotic cell to do this, because only a mitotic cell will have chromosomes that are what? Condensed. Very good. Only a mitotic cell will have chromosomes that are visible, that are condensed, that you can see, and then you can spread them out and you can study them, right? So we're going to look at a mitotic cell with the chromosomes all spread out. It's called a karyotype, right? Karyotype is a spread out composition of mitotic chromosomes. All right? Maybe you're thinking, I thought we were talking about meiosis. Why are you talking about mitosis? It doesn't matter. It could be meiosis as well. The point is the chromosomes are condensed, which I hope you agree is what you're seeing here. Okay, this is the photograph now of the human karyotype. All right, so what are we seeing? Yeah, so first thing to notice is chromosomes can be different sizes. This is certainly true for human chromosomes, right? We have big chromosomes, we have medium chromosomes, we have little chromosomes, and this, our human karyotype, I think, adequately shows that variations in size that can happen. Earlier in the lecture, we said there's 46 chromosomes in human cells. Right? We said 46. Let's see if that's correct. Okay, so we said this is a human karyotype. The content of a human cell is this 46 chromosomes. Now, let's see. 1, 2. With a box around it for some reason. 3, 4, 5, 6, 7, 8, 9, 10. Okay, 10 chromosomes per row, so that would be 20. Yes? Okay, 30, and now it's getting a little awkward. Okay, 31, 32, 33, 34, 35, 36, 37, 38, different size here. 39, 40, 41, 42, 43, 44, 45, 46. It is 46. Yes. So, it makes sense, right? It's a human karyotype, it must be 46 chromosomes. And so it is. Okay. Please recognize they are replicated chromosomes. What did we say? Every cell division preceded by what? Unit replication. Very good. So, that's what's happening, right? This is a cell division that's happening, mitosis, meiosis, whatever. The chromosomes have been replicated. Now, this is not obvious, I think. Don't just listen and you know, maybe I'm lying, right? Skepticism, right? Are they really replicated? They're replicated. Well, let's see. This is not obvious to me. All right, I'm sorry. This is not obviously a replicated chromosome. You gotta look at it, I think, to believe this. What about, okay, I'm gonna come way down here. What about this? Is that two sister chromatids? Can you convince yourself that's two sister chromatids? What about, I don't know, here? How about that? Is that two sister chromatids? It's hard to see, but they are replicated chromosomes. Yes. And clearly the authors of the textbook realized that a karyotype of humans doesn't necessarily obviously show replicated chromosomes, so they drew it. They drew it! So you can prove to yourself these are replicated chromosome. That's a replicated chromosome. There it is. There it is. A replicated chromosome contains two sister chromatids. There they are. You can see them in the cartoon. And there are the labels. Okay? Everyone see this? It's difficult. Because they're so long is what makes it difficult, I think. But just, okay. Yeah, good? Okay. Also notice that they're paired. I mean, this is the elephant in the room, I think, right? The chromosomes have been paired, right? There's even a big box around the first pair. What is that? Okay, so this now we need to talk about, because the pairs, right? They're all paired, right? Aren't they all, almost all of them? Yeah, every one of them, even the funny pair here. They're all paired. These pairs are chromosomes that have the same genes. That's why they've been paired. The chromosomes have the same genes. And then we have a name for them. I mean, we've got that name. Chromosomes with the same genes. They're called homologous, right? Same. Homo, right? Homologous. So homologous chromosomes in sexually reproducing species are the pairs of chromosomes with the same genes. Why do we have homologous chromosomes with the same genes? Because we have two parents. Yeah, it's sexual reproduction. Yes, each parent gives us one of each chromosome. And so we have two with the same genes. Okay? So So one of each pair is inherited from each parent. You have a maternal chromosome, a paternal chromosome. Next pair, you have a maternal chromosome, you have a paternal chromosome. Next pair, maternal chromosome, paternal chromosome. What's the implication? Well, I guess how many of each gene would you have? Of each gene, you have 2 copies, right? A maternal and a paternal. That's exactly what the implication is. Each parent is giving you 1 copy of a gene, you have 2. How many insulin genes do you have? Insulin is a protein. It's included by the insulin gene. How many insulin genes do you have? 2 insulin genes. 1 from your mom, 1 from your dad. Hemoglobin, it's a protein. Carries oxygen in the blood. How many hemoglobin genes do you have? You have 2, right? 1 from your mom, 1 from your dad. Yeah, okay, good. So how does meiosis half the number of chromosomes? Right? What is that about? Simple. We separate the pairs. We separate the homologous pairs. Meiosis separates the homologous pairs. That's the whole strategy. You have 2 of each chromosome. Separate them. So the gametes will only have 1. 1 of each chromosome. Yeah, big concept. That's a big concept too. Okay, big concept. Question? Sorry, question? Okay, although homologous chromosomes have the same genes, please understand they're often not identical. Okay, what does that mean? Okay, so for example, I looked it up. The gene for eye color, human eye color, is on chromosome number 15. So if you didn't notice earlier, the chromosomes are numbered. The numbering is also showing the karyotype. So chromosome 15 here, which you have 2 of, carries the eye color gene, right? What do I mean? The genes are not identical? Well, for example, maybe the maybe the eye color gene within the chromosome on the left is for brown eye color, whereas the eye color gene in the chromosome on the right is for blue eye color. You see that? And then what is the eye color of the offspring, blue or brown? I don't know. I'm just saying. There's something will happen, right? Something, but you get the idea, right? So we need a name for that, right? Well, we're basically recognizing that genes can have variations, right? Genes themselves can have variations. The eye color gene can have variations. And the variations of a gene are called what? Alleles, right? That's for a name, right? Alleles. They're called alleles of a gene. The variations. And we can now specify in 2026 in chemical terms what are alleles of a gene. They are genes that differ slightly in what? Sequence of nucleotides is one answer. Base pairs, I think, is another one. Sure. Slight differences in their nucleotide sequence. This is the term, remember, let's see, for the monomers that make up DNA and RNA, nucleotide, the DNA and RNA polymers. So the alleles of the gene would have slightly different nucleotide sequences. Okay? Okay? All right. So be careful with the terminology again. And in this case, you got to watch out homologous chromosomes, sister chromatids, because they're different things, aren't they? And then this is where you could. I told you earlier you got to remember DNA replication happens before cell division. Well, you should also remember not to confuse homologous chromosomes and sister chromatids. Now, which term refers to 2 things that are identical? identical, 100% the same. Sister chromatids. Yeah. Sister chromatids, remember, are the products of DNA replication. That's the DNA copying mechanism we spoke of. And that gives you identical DNAs. Okay, so which are identical? Sister chromatid. And again, that's just something that happens before every cell division. You make sister chromatids. All right? Now, which of those two are present throughout the entire cell cycle? It's only one of two. Present throughout the cell cycle. There's a big thing mitosis now, right? In your cell. Mitosis. Mitosis. Which of the two are present throughout that cycle? Okay, if you said sister chromatid, be careful. Okay, sorry. But remember, why do cells do DNA replication? In order to then do cell division, right? It's a preparation for cell division. Okay, so that immediately implies that sister chromatid is not always there. It's a preparation for cell division. But the cell is not dividing, and not going to. Well, don't do DNA replication. Oh gosh, remember your cell cycle, guys. Connect that. So sister chromatid is not always there. They're not there in G1. They appear in S phase, and then they're there in G2, and then they're pulled apart in N phase. Okay. Homologous chromosomes are always there. Right? How many do you have, each of your cells? 46. Right. 23 pairs, 46 chromosomes. Always there. Now, unless you do meiosis, right? That's a different story. We're not talking about that. Okay? Cell cycle. Question? You can see the gears turning. Okay, all right, think about it. So yeah, I just put this in here so we can just make the point. Again, G1, there's no sister chromatids. DNA replication happens in S phase. It gives us sister chromatids, which are then pulled apart in cell division, right? In contrast, homologous chromosomes are here throughout the cycle. They're not going anywhere, right? Unless you do meiosis, which is a special case. Okay? All right. What's the deal with the big chromosome and the small chromosome? Talk about that.
Sex chromosomes.
Okay, so remember that there are particular chromosomes involved in determining sex, right? Sex, male or female, right? So two chromosomes in mammals are going to determine sex. We just call those sex chromosomes, right? Pretty simple. Then you need a name for all the other chromosomes, right? So we'll call those autosomes. All the chromosomes are autosomes except for the two sex chromosomes. So for example, humans have 44 autosomes and 2 sex chromosomes, right? And was this a male or a female? What was the male? Okay, good, right? Because males have one of each, don't they? So please be aware, human males have an X and a Y. Human females, 2 X's. You do that. Be aware, X and Y not homologous chromosomes. What do we say homologous chromosomes are? They have the same genes. X and Y don't have that. Not homologous. You know this. One is like that, and one is like that. How do they have the same genes? So they have different genes. Let's just say that. X and Y, different genes. How many homologous pairs do human males have? How many homologous pairs of chromosomes do males have? 22. Very good. How many do females have? 23. Very nice. All right. Any questions about chromosomes at this point? All right. very good. I know it's a big transition from glucose and glycolysis, but stay on it, okay? I'll be in my office tomorrow. If you open the door to his office right now.